Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speakers your questions. Your questions in the question and answer window will only be visible to myself and our speakers. So thank you for joining us today. It is now my pleasure to introduce our speakers, Jason Sterling and John Rockwell from Catalent Biologics.
Hey, thank you for that wonderful introduction. Today we'll be exploring new and improved analytical methods for traditional and unique modalities. Here's a brief agenda of the talk we'll be giving today. We'll provide an overview of Catalent and our capabilities. We'll discuss some capabilities of the Biologics Analytical Services team. I'll be presenting some results that we've collected on Sialic and Glycans. Then I'll be handing off the talk to John, who will discuss E&L analysis for ADC free drug assays. And we'll conclude the whole talk at the end of that. So Catalent is the world's leading partner for pharmaceutical, biologic, and consumer health innovators in development, delivery technologies, clinical logistics, as well as clinical and commercial manufacturing across the widest spectrum of technologies and dose forms. Catalent is a public company listed in the New York Stock Exchange with over $2.7 billion revenues annually and 13,000 team members around the world. Here is a listing of all of our capabilities in the Catalent Biologics network. These capabilities provide our customers with full lifecycle support from discovery to commercial phase and beyond, and on a variety of different types of products. Our sister sites allow us to provide a variety of drug development services to our customers. You can see from this list that each of the sites have unique capabilities or shared capabilities depending on the site. We can do everything from cell line development to drug substance process development to drug product formulation and fill finishing. This allows Catalent to provide an enormous amount of flexibility into any drug development pipeline. Today we will be focusing on some of the work being done at two of these sites, the Morrisville RTP site in North Carolina and the Kansas City site in Missouri. So let's move on to the BAS, or Biologics Analytical Services Group. This group spans across multiple sites within the Catalent network as well. The BAS group specializes in providing lifecycle support for all of your biologics needs, including method development, method validation, stability testing, and extractable releasables testing, as well as biophysical characterization. Specifically, at the Kansas City and RTP sites, we specialize in biophysical characterization, stability testing, and the development of methods to measure compound CQAs. Some unique strengths of the Kansas City site are the BLI and SPR testing, analytical ultracentrifugation testing, FACS analysis, the analysis of polysorbates and other kinds of lipid molecules, and the characterization and testing of ADCs. Unique strengths of the RTP site are that we are fully cGMP and cGLP compliant, even our mass spectrometry space. We have an automation group, and we also have a radioisotope lab that can be used to do specific types of testing. I'd now like to move on and talk about some work that we've done in RTP, in our Morrisville site, on the analysis of Sialic and Glycans. So sialic acids are an ancient family of 9-carbon monosaccharides, or non-ulosonic acids, that are found in eubacteria, archaea, and the Deuterostomia superphylum, which includes humans, actually. Here I'm only showing the two most commonly studied sialic acids on this slide, and Nu-5-Ac and Nu-5-GC, which are N-acetylnumeric acid or N-glycolyl numeric acid. Numerous additional forms of these two sugars exist in nature, in which the monosaccharides can be further modified by phosphorylation, sulfation, lactylation, acetylation, and methylation. Taking these additional chemical substitutions into consideration, there may be upwards of about 50 different distinct N-acetyl uramic acid monosaccharide structures that could be detected analytically in nature, making it one of the most heterogeneous sugars that we can ever detect or analyze. Sialic acids are also found as terminal monosaccharides on N-Glycans, O-glycans, and glycosphingolipids. They also cap side chains of GPI-anchored proteins in vertebrates. And they've been found actually as an internal sugar in some bacterial and invertebrate polysaccharides. They are the predominant sugar on all cell surfaces, forming a complex sialome on the cell surface, which is integral to a number of cellular and biological functions. These include cell-to-cell interaction, recognition, and signaling, functioning as a cytoprotector. They have a strong role in immunology and protein stability, in ion binding and transport. They've been found to be implicated in cancer metastasis, metabolism, as well as the infection of pathogens, and even in human evolution. So it's no wonder that we want to study these very complicated and interesting molecules. Today I'm going to focus on sialic acids attached to N-Glycans. This is an idealized structure of a complex N-Glycan. It has a core structure of monosaccharides, it's glycosylated, and it's also been modified by Golgi-resident glycosytransferases to contain LUCNAC, beta-length galactose, and terminal sialic acid residues. These terminal residues can be linked alpha-2,3 or alpha-2,6 on N-Glycans. We can also see alpha-2,6 linked sialic acid to GalNAc, and we've also been able to discover the creation of alpha-2,8 or alpha-2,9 linkages to other sialic acids to form polysialic acid chains in specific cell types. But I digress. We're going to focus on N-Glycans a little bit. So studying N-Glycan sialic acids can be somewhat tricky, as these sugars don't absorb light or fluoresce naturally. So in addition to the sialic acid residues being attached to N-Glycans, there's a problem with them in that they're labile. And so if you were going to do any kind of mass spectrometry analysis on them, they can fall off in the source or during its transport through the flight tube. In addition, sialated N-Glycans don't ionize as well in positive ion mode as their neutral N-Glycan counterparts, and the negative charge can result in strong addict formation when you're looking at these molecules in positive mode. So nevertheless, a number of methods have been developed, mass spectrometry methods and HPLC-based methods, to study these incredibly interesting structures. We can use, for example, intact mass analysis, glycopeptide mapping, N-Glycan profiling, and sialic acid analysis. There's another way that I've discovered and that we're trying to work with right now and implement in our labs that you can study these N-Glycans, these complex sugars that I'm discussed here and won't be discussing today. And that's a derivatization technique that was developed about a decade ago that allows one to differentiate between terminal alpha-2,3 or alpha-2,6 linked sialic acid. I will not be talking about this technique nor the intact mass analysis results today due to the confidential nature of some of these results that we obtained. So I'll be talking about results that I've collected on a model glycoprotein that we've worked on in our lab. It's a non-IgG-like glycoprotein in preclinical development. It's about 35,000 Daltons in size, and it has 2 N-glycosylation sites on it. First, I'd like to talk about how we perform glycopeptide mapping analysis on this particular protein. So a typical experiment will look like this. There's a kind of a schematic. We start off with our glycoprotein sample. We'll mix that sample with a zinc buffer containing guanidine. We'll then reduce the protein's specific bonds with BTT and alkylate all the free cysteine residues with acetamide. We can remove the excess guanidine using a dissolving column, and then this reduced alkylated and buffer exchanged protein is then digested with a protease, We lose trypsin a lot, but also lyc, aspen, and gluc. And we'll do this for a specified amount of time, and the reaction will be split into two different aliquots. The first aliquot you see in the top will be quenched right away with acid and will become our glycopeptide mapping sample. The second aliquot will be treated with P&J-F, which is a peptide N-Glycanase F. It's an enzyme that's used to cleave N-Glycans from polypeptide chains. We will then quench the second reaction after it's been treated with P&J-F, and both samples will be analyzed together at different times using the reverse phase UPLC SMS technique that we developed in-house. The results of a typical experiment look like this, where we're looking at on the y-axis ion counts, on the x-axis the time in the chromatogram. So this slide shows a mirror image of these results from a peptide map on the top, a glycopeptide map on the bottom from the same sample. We can see that the mirror TICs, our total ion chromatograms, look very similar to each other. So all the major peptides that are present in the sample are present in both aliquots that we tested. But if we look closer, in the circled areas I show on the slide, there are some differences here where I call them P1 or region P1 and region P2. And these correspond to the peptides, glycopeptides, that elute on this chromatogram that are corresponding to the N-glycosylated sites on the glycoprotein that we have. So this is where the glycopeptides would elute as well as their non-glycosylated counterparts. So the peaks that are present in the glycopeptide map but absent in the peptide map are the glycopeptides, and you see quite a few of them in both those regions. When we analyze the masses of these structures and convert those to N-glycan structures, because we know the peptide mass, it allows us to compare the type and relative amounts of each N-glycan present at each N-glycosygen site. The results, when you compile them, look like this. So what we're showing are the compiled results from testing two lots, and this is the glycopeptides detected at the region P1 of this protein. It's composed mainly of dientenary neutral N-glycans that terminate in mannose, glycnac, or galactose. We do see some solid N-glycan structures, but they're at very low levels compared to the other N-glycans that attach to this site. The most abundant N-glycan is the one terminating in glycnac here at the top, and it's known as F6A2, or commonly known as G0F. This site has a relatively low percentage, as I said, of solid N-glycans, and this result's consistent between the two lots that we tested. When we look at the other region of the site that was glycosylated on this protein, region P2, we see that it has a much higher percentage of N-glycans terminating in sialic acid. In addition, the neutral and solid N-glycans that we do detect are way more mature from a biochemical sense than those N-glycans detected at site P1. So we see a lot of dientenary as well as trientenary N-glycans that terminate in mannose, glycnac, galactose, and N-acetyl neurominic acid. The most abundant structures at this site were F6A2G2 or G2F, commonly known as G2F, and F6A2G2S1, which is the one more in the middle of the slide and has one solid structure, and that was more commonly known as G2FS1. So I've highlighted a couple of other structures here that you can see on the bottom of the slide in blue squares. And these are N-glycans that have acylated N-acetyl neurominic acid. This indicates that the method is capable of detecting these acylated sialic acids, which is a kind of a nice feature to have for this kind of analysis. So the difference between the number and type of structures at each site, so comparing site 1 and site 2, you can use some of the information from these N-glycan structures at both sites to infer some details about the 3D structure of this glycoprotein. So for example, we can see that site P2 is dominated by more mature complex N-glycan structures compared to site P1. This suggests that P2 may be more surface exposed than P1, or conversely that P1 may be more sterically hindered than P2. And indeed, P1 corresponds to the conserved N-glycan site found on all IgG1s, the FC region that you'll see. It is well known that this conserved site is sterically hindered. They've done 3D crystallography studies on that to prove that. and that prevents some Golgi resident glycosyl from getting into that site and modifying those N-Glycan structures at that site into more mature structures. That's why you'll see more diantennary, less solid structures at that site compared to site P2, which may be more surface exposed. So glycopeptide mapping can provide some site-specific details on the N-Glycans at each site, and it can also be used to delve into the 3D structure of the protein you're studying. Next, I'll move on to N-Glycan profiling, which is the most common form of analysis that's done on N-Glycans in labs like mine. So this slide shows our experimental procedure for N-Glycan analysis. We use the Waters Rapid Flow reagent for this analysis. We use a couple of other types of tests as well, 2A labeling, 2AB labeling, as well as a label from Agilent. However, this results I'm talking about today are from the Waters Rapid Flow MS procedure. So we start with the glycoprotein, and we denature it using the Waters Rapid Jet reagent. We add P and JZF to remove the N-Glycans from the glycoprotein, and then derivatize the released N-Glycans using this Rapid Flow MS reagent. We quench the reaction by diluting with acetonitrile, and then we use a Sialic solid phase extraction plate to remove the excess Rapid Flow reagent. The eluted N-Glycans that are derivatized now, we can then analyze them directly on an amide column attached to our UPLC system. We can use MS or MSMS analysis for that type of test. So a typical result will look like this. We can see that this is a fluorescence chromatogram. It shows representative N-Glycan structures, representative N-Glycan profiling result. You can see the y-axis contains or displays fluorescence, while the x-axis shows the retention time of each N-Glycan. We can identify these N-Glycans using the mass of the actual N-Glycan, the fragmentation of the N-Glycan, as well as another property called glycan units, which correlates standard derivatized glycan chains to retention times. And so by having this standardization process, you can assign a new value to each N-Glycan that can help infer its structure. And that's the benefit of using GUs for the identity of some of these N-Glycans. You can identify isobaric structures that have the same mass and same fragmentation patterns, or relatively the same fragmentation patterns, but separate in time or by chromatography. And you can't figure out the structure is unless you do a more high-level analysis. With the GUs, you can assign a structure that has already been fully elucidated by other scientists, and that GU unit will actually assign that structures where the sugar, terminal sugar, is located in each N-Glycan chain. So one thing we noticed right away is that the number of structures we detect in this glycopeptide mapping experiment the number of structures that we detect in this N-Glycan profiling experiment differ a little bit from the number of N-Glycans that we detected in the glycopeptide mapping results I showed earlier. There's less structures. And that's because the N-Glycan profiling experiment provides a relative quantitation of the N-Glycans observed at both sites. It turns out that site P2 is only occupied about half the time on this glycoprotein. And so that means that the P2 N-Glycans are unrepresented in N-Glycan profiling results. And so their overall relative abundances will be a little bit lower than those from P1. When you compile the results from that experiment into a table, and what I'm looking at here are summarized N-Glycan profiling results from 2 lots of this IgG-like glycoprotein. We can look at the relative abundances of each structure that we detected using in the previous slide. And so to make this data set a little easier, I've combined the abundances of isobaric structures that we detected in the previous slide into one structure shown here. So we routinely perform these types of lot-to-lot comparisons for our customers to help them determine the effect of upstream and downstream optimization experiments on their proteins and N-Glycan profile. And so N-Glycan profiling data, one of the benefits of it is that you can generate this data in a couple of days, making it much easier to perform than glycopeptide mapping studies. Finally, I'd like to talk about another type of analysis that we do in our labs, and it's the only method that is quantitative for sialic acid. So sialic acid analysis, as it's most commonly called, is a method that gives you quantitative results for sialic acid from your target glycoprotein. So glycopeptide mapping and N-Glycan profiling only provide a relative quantitation of each solid structure, not an absolute value. And that can be a little bit tricky, as the results from glycopeptide mapping, N-Glycan profiling, and even Sialic acid analysis may not correlate as well. But the one thing to remember is that this particular analysis is the only one of the three that's quantitative. The other two are great for looking at overall global changes to N-Glycan profiles, depending on changes that are being done to the process for making that particular protein. So a typical Sialic acid experiment starts by incubating a glycoprotein in mild acid hydrolysis conditions. This will cleave off the Sialic acids from the terminal positions of the N-Glycan. The released Sialic acids are then derivatized with a fluorescent compound like DMB. We'll then purify those derivatized compounds and analyze those Sialic acids by FLD-HPLC. If you do this with standards, you can actually create a nice standard curve. So we take the same process I described in the previous slide. We'll take some Sialic acid standards. In this case, I'm only showing results from N-glycolyl nuramic acid, N-glycolyl nuramic acid, and we can create these nice standard curves where you're measuring the amount of fluorescence you obtain per picomole amount of nuramic acid injected onto the HPLC column. So these standard curves are really good. You can see that this correlation curve that we have for both these molecules have an R-squared of 1. And so with these curves, we can then take any sample, any unknown glycoprotein sample, put it through the same process, and then have a measurement of how much picomole amount of Sialic acid is present from that sample. So results from that experiment are not as pretty as what you saw in earlier slides, but you do get a nice little table that's very easy to see. A chromatogram is showing in the foreground. It shows retention time on the x-axis versus fluorescence intensity on the y-axis. And there's an inset, the table in the inset there shows the results for 2 lots of the IgG-like glycoprotein, looking at the presence of new 5GC and 5AC. And we can see that we didn't see any 5GC at all in this particular sample, but there's 1.1 picomoles of mole per mole basis, 1.1 new 5AC inside this, attached to this particular protein here. So in conclusion, glycopeptide mapping provides some site-specific N-Glycan results that you can't get from the other 2 techniques. You can use this results to provide limited structural information of your protein, but it's difficult to quantitate solid species because of coelution and because it's very difficult to obtain quantitative results because of that. The next analysis that we did was N-Glycan glycoprofiling, where you provide a profile of all the different N-Glycans attached to both sites or any number of sites you have on your molecule. You can incorporate additional steps to this process very easily. You can do exoglycosidase digestions or derivatizations of the terminal sugars to show their orientation. And the results are dependent on the overall N-Glycan abundance which is in contrast to what you get with glycopeptide mapping. And finally, I showed results of the Sialic acid analysis, which is the only quantitative method that's out there right now for Sialic acids, but it doesn't provide any information on the N-Glycan structures from which those particular Sialic acids were derived from. And with that, I'd like to hand off the talk to John, who will then talk about work he's done in his lab.
Thank you, Jason. Switching gears, we're going to talk a little bit about some work that's been done in biologics at Kansas City in light of some analytical challenges we've observed with free drug analysis during antibody drug conjugate development. I wanted to start with an overview of the structure and function of an antibody drug conjugate, or ADC. Generally speaking, the purpose of an ADC is to deliver targeted toxin payload to a malignancy rather than blasting the patient with chemo. Toxin is covalently bonded to an antibody using a linker structure. Usually there are several payloads linked to a single antibody. And depending on the indication, the antibody will have a target affinity for some moiety on a malignancy. Shown here is the structure of Gemtuzumab, ozogamicin, marketed previously as Mylotarg. For Mylotarg, by linking calicamycin, which is part of a class of cytotoxic antibiotics, to anti-CD33, it can be delivered to a leukemic stem cell expressing CD33. This product was first marketed, it's the first marketed ADC approved by the FDA back in 2000, and it was marketed as a second-line treatment for acute myeloid leukemia, or AML. Note that the payload for the free drug itself is, in many cases, the actual first-line treatment for that indication. This slide, I just wanted to go over as an overview of extractables and leachables. Extractables are compounds that are released from product container closure, packaging, or delivery systems under accelerated conditions. For example, heat, photoirradiation, or maybe in the presence of strong solvents. And leachables are compounds that are released from product container closure, packaging, or delivery systems under normal conditions, or the conditions that you would normally store the product over the lifetime of the product. Leachables are typically a subset of extractables, and formal extractables and leachable studies are normally conducted as part of pharmaceutical development, following the guidance from the FDA on container closure systems for packaging human drugs and biologics issued in May of 1999. The scope of this presentation, or the remainder of it, relates to the extractables and leachables from Labware used to store and test ADC drug substance and product. Testing of free drug toxin levels in ADCs is performed during release and stability to ensure that it remains below safe levels. Polymeric labware that is typically considered acceptable for the storage and handling of biologics can leach compounds that have similar hydrophobicity and UV absorbance to that of the free drug. And an exploration of compatible containers that are safe for storage of biologics and that do not contribute peaks to the residual free drug assays is prudent. This is an overview of Labware polymer manufacturing. This is just scratching the surface. I don't know the details on the actual processes, but in general, manufacturing of polymers for lab use involves several steps to transform the natural resource to its final product. The monomeric compounds in petroleum are refined and processed to form polymers. Polymers are further processed with additives to make them suitable for storage of reagents and drugs. Finally, the end products are cast using lubricants and solvents and then distributed to the end user, which in this case is labs and pharmaceutical manufacturers. Each manufacturer may use unique processing agents in the manufacture of their plastics, and this can lead to a different set of extractables and leachables. The extractables and leachables from polymers may be completely inert and not impact product stability, but can present a problem for certain testing, as will be shown later. A case study was performed to evaluate various polymers that an ADC may come in contact with during storage and testing. The goal was to determine if any of the labware contributes peaks that interfere in a reverse phase assay used to measure free drug levels. There were two different solvents that we selected. One was ultra-pure water, and the other was a histidine buffer containing surfactant at neutral pH. This buffer system is very common formulation for biologics, which is why it was selected. I just want to note that the water was selected as a control for the buffer in case we saw extra peaks that originate from the buffer. We wanted to be sure that that was the case and not something else leaching from the containers. Both solvents were prepared and stored in glass media bottles that had been previously rinsed with methanol and water. The interaction of each of these two solvents with various containers and pipettes were evaluated. Labware included glass pipettes, polypropylene pipettes, 10R glass vials with ethylene tetrafluoroethylene coated stoppers, and we both did upright and inverted for that vial. Also for the 5 ml polyethylene terephthalate glycol bottles, or PETG bottles, we also did those upright and inverted to test whether or not there's something that might leach from the caps separately. And then we also evaluated 5 different polypropylene cryovials from different vendors. The pipette samples were prepared by aspirating and dispensing solvent a single time into glass chromatography vials for analysis. The 10R glass vials, PETG bottles, and cryovials were prepared and exposed to several extractable and leachable conditions. We did 40 degrees Celsius for 6 hours as an extractable condition. Remember, the extractable is a condition that your product would not normally see during its shelf life. And then 2 leachable conditions. One was minus 80 degrees Celsius and the other was 5 degrees Celsius, both for 2 weeks. Note that the conditions selected for the study are very mild compared to common stability conditions required for biologics development. Typical stability studies extend out to 3 months for 40 degrees C and up to several years for minus 80 and 5 degrees. Each labware and condition was prepared in triplicate. After exposure, the samples were transferred to glass chromatography vials for analysis. Analysis was performed with a single injection of each replicate using a C8 column with the water acetonitrile gradient containing trifluoroacetic acid. Controllers included 10 nanogram free drug standard and then both of the solvents from the source bottles. Now, the controls in this slide here are overlaid to show that there are no interfering peaks that leach from the glass or the polypropylene pipette tips, at least in the quantifiable range of this method. And for this method, that's mostly what we care about, the range from about 4 to 23 minutes. The peaks appearing from about 10 to 22 minutes in the 10 nanogram free drug peak injection are free drug peaks and free drug related substances. There are peaks that appear in the free drug and buffer controls that are not present in the water, but we believe those are related to the buffer components. And they don't appear to interfere in the region where we want to quantitate free drug or related peaks. The purpose of using water was, again, to just control for potential buffer peaks. Biologics would rarely be formulated in just water. Since there's no peak contribution from the buffer controls from 4 to 23 minutes, the buffer and the remainder of the slides is going to be used for display purposes. The slide also demonstrates that there is no contribution from the chromatography vials used for all of the analyses. This is an overlay of buffer in the 10R glass vials and the 5 ml PETG bottles, both inverted and upright, after 6 hours at 40 degrees Celsius. The other leachable conditions were not evaluated because they also showed no interference in the profile. This is the most extreme condition, and since there's no apparent extractable peaks that interfere, we're comfortable just showing these. The containers, the glass vials are popular containers for drug product storage, and the PETG bottles are popular storage containers for drug substance. These would likely be a part of a formal extractable leachable study, depending on the development of that product. But it's good to see here that there's no interference. This is an overlay of mild leachable conditions of buffer for 2 weeks at minus 80 degrees C in 5 different cryovials represented as cryo A through E. This condition is a conservative long-term storage temperature for biologics. Cryo A, B, and E show leachable peaks highlighted with the red circles. It's interesting to note that although cryo A and B are marketed by different manufacturers, the leachable peaks in the profile appear very similar. It's likely these share similar manufacturing processes. Cryo-E is unique with a series of leachable peaks that elute from about 4 to 7 minutes. Keep in mind that leachables shown here were generated after very limited exposure to the vials. There was probably about an hour of time after pipetting the buffer into the vials until it was frozen, and then again once we pulled it out after 2 weeks, it was thawed and transferred to LC vials. So maybe 2 to 3 hours tops exposure to those vials, at least in liquid form. The overlays on this slide show moderate leachable conditions from buffer in cryovials after 2 weeks at 5 degrees Celsius. 5C is a typical short-term storage condition for biologics. Here we see that cryo A and B show similar leachable conditions as the -80 in the previous slide. Cryo C and D show small peaks corresponding to the largest peaks in vial A and B at about 13 minutes. Cryo E has elevated peak levels from 4 to 7 minutes and the formation of new peaks at 10 and 12 minutes. And here, finally, we have extractable conditions for at 40 degrees C for 6 hours. This temperature is commonly used during stability studies for up to 3 months as an accelerated condition and is used to predict molecule stability over a longer period of time at lower temperature storage. Extraction shows significant increases in peak intensities for vial A and B. Vials D and E show modest increases in the same peaks seen at 5 degrees C. And cryo C, which is interesting, has a clean profile, given that the 5 degrees C for 2 weeks profile did leach a small peak at about 13 minutes. And that could be either a result of variability in the leachables from vial to vial or a greater dependence on the length of time of exposure rather than the thermal energy. None of the cryo C replicates showed any peaks for this condition, both in buffer or with water alone. So using free drug as a standard, all the extractable leachable peaks in the cryovials were quantitated. The bars represent the equivalent quantity of free drug based on the total peak area of the extractable leachables. And the blue bar is the -80 C, the purple bar is the 5 C, and the red bar is the 40 degrees C condition. The chart shows also an example specification limit of at 40 nanograms of free drug. The 40 C conditions for vials A and B already exceed that limit without the addition of an 80 C drug substance or product into the profile. 80 C substance or product will likely contain a certain amount of endogenous free drug, either as a residual impurity or a stress-induced product. And cryo C and D, or even E for that matter, would be a clear choice over A and B for 80 C sample storage. So what are those peaks? We first tried to collect some spectra using a diode array detector, but there wasn't enough signal to get a distinguishable spectrum from any of those peaks. Even the largest peak, we couldn't get anything meaningful out of that. The largest peak in the profile with a retention time of about 13 minutes was the only peak with a strong enough mass spec signal to obtain a mass. And that peak had a mass matching the formula C9H11O. Due to the many possible structures with that formula, it's hard to say what it is, but a brief search on PubChem yielded 146 unique structures with that formula. I dug into some patent uses for likely structures, and those included a biodegradable solvent, an ingredient for moisture and oxygen barrier in plastics, and an ingredient for a UV protectant in plastic. In a situation where leaching is already a contaminated source, it's possible to perform a large-scale extraction and concentrate the compounds to improve the abundance for a positive identification. Another option might include nuclear magnetic resonance. And it would be nice to know the peak IDs of all these peaks from a scientific standpoint, but something that really we want to try to avoid. It's not necessary if we minimize the leaching of those peaks by selecting a proper vial. Okay, the conclusions for this are typical drug substance and product storage does not appear to be the source of extractables and leachables that would interfere with free drug analysis, which is good. Attempt at characterizing these compounds can be challenging and probably best avoided by proper vial selection. This has to be done empirically to determine compatibility based on the specific free drug assay that would be used in routine analysis. Glass does not appear to leach anything at all and would be a safe alternative to polypropylene cryovials if you wanted to take a conservative approach. But that would need to be evaluated whether or not glass storage is appropriate for the storage of your sample, especially if you're going down to -80 degrees C. So with that concludes our talk for today, and we'd be happy to take any questions.
Okay, great, thanks. So the first question is, what are some other challenges besides extractables in ADC development?
Sure. So since the challenge, I think, in general with an ADC is that a lot of biologics labs are contracted or undergo the development of ADCs, mainly because the largest structure in the molecule is a biologic. But it essentially is 3 molecules bundled into one program, at least for analytics, because you have the free drug with the linker, you have the monoclonal, and then you have the combination, the conjugate. And each of those require a certain amount of characterization. So another thing too, and maybe I should have started with safety first, right? The free drug used in the conjugate it's highly toxic. Like I said before, it belongs to a class of cytotoxic antibiotics, and in powder form, it can become aerosolized and is highly recommended to handle that inside of a glove box to protect the analyst. And even in liquid form, a lot of the solvents used to solubilize these for analysis, for example, DMSO, actually enhance the ability of the toxin to pass through your membranes, through your skin, whatever it may come in contact with. Additionally, it's a challenge for other assays like size-based methods, where you have a heterogeneous mixture of ADCs, not in the sense that you have, on top of the antibody heterogeneities that you may normally encounter, you also have a varying number of free drug that is bonded to the antibody, and depending on how many of those are bonded, it changes the properties of that molecule, such as for UV concentration measurement, or like I said, any size-based method like size exclusion or CESDS. So there definitely are some analytical challenges. I can't comment on the process challenges, although I'm sure there are some there as well. But analytically, there's a lot to consider with ADCs that we don't normally have to consider for just a typical MAB. Thank you.
Okay. And given the many species of free drug that elute in the profile, is it possible that there are additional free drug species that elute outside the window that are unaccounted for?
That's a great question, and it's one that it would go into the development of the method. There would need to be some evaluation during method development whether you think you're getting all of the peaks accounted for. A great way to do that, and it would also be a part of method validation, which would be to do a spiking and recovery experiment where you spike in free drug to your sample, and then determine an acceptable level of recovery to ensure that you're counting all of the possible free drug species, because there can be many adducts that form. The linker is it's highly labile, and if that for whatever reason falls off the antibody, it's going to be reactive with something else, or the free drug itself maybe bonding to components of the formulation. So yeah, but as part of the method development, an evaluation of whether or not you can spike and recover the free drug to make sure that you're counting everything as prudent.
Great. Thanks, Jason and John. And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.